Good morning, everyone, and to those of you also that are listening in on the telemedicine channel. Thank you. We are particularly honored this week, and as you know, this is our Research and Innovation Week, and the Dr. Bradshaw, the awards are this afternoon at 2:30. We just had an amazing group of speakers, but I'm particularly excited about all of the posters and the research that has been shown and demonstrated in the engagement all across the health system. I'm honored today to have the opportunity to introduce Dr. Stephen Rosenberg. Steve is Chief of Surgery at the National Cancer Institute in Bethesda, Maryland, and a professor of surgery at the Uniformed Services University of Health Sciences, as well as the George Washington University School of Medicine and Health Sciences. So we're glad we're all on the, I can now say I'm on the same faculty, which is a great honor for me. He got his academic start at Johns Hopkins with his undergraduate and MD degrees, and then a PhD in biophysics at Harvard University. One of the things I like most about Dr. Rosenberg's CV is that he and I trained at the same hospital and surgery in Boston, the Peter Bent Brigham Hospital. It's now known as the Brigham and Women's Hospital, but it was one of the premier training programs. And when he started, he was in a class of interns that became known, and for good reason, but became known as the Magnificent Seven. And at least two, maybe three of the surgeons in that group were pediatric surgeons. And one was the father of ECMO as well as two leaders, as well as two other leaders in pediatric surgery that ran a big-time training program. He became the chief of surgery at the NCI and the NIH, and he is still there and still in that role. And he developed the first, and really the reason we're all here today to hear about this, the first effective immunotherapies and gene therapies for patients with advanced cancer. And that may seem very routine now and part of everything we do, but at the time that he started this, it was revolutionary. There were people that probably thought he was crazy for thinking that the immune system had any role in fighting cancer. His studies of cell transfer immunotherapy have resulted in durable and complete remissions in patients with metastatic melanoma, for example. And he was the first to successfully insert foreign genes into humans. And his recent studies, which we will hear about, of the adoptive transfer of genetically modified lymphocytes has resulted in the regression of metastatic cancer in patients with melanoma, sarcomas, and lymphomas. So just think about that. These were conditions, situations that were just thought that there would never be effective therapy about that. So because of that, he is widely known, and certainly by me, as the father of T-cell therapy, just like Dr. Thomas is known as the father of bone marrow transplant. So you can think about all the impact that Dr. Rosenberg has had. So therefore, it is a huge honor that he is here to speak with us today on cell transfer immunotherapy for common epithelial cancers. Please welcome Dr. Stephen Rosenberg.
Well, it is a pleasure and honor to be here at one of the world's great children's medical centers. And I'm delighted to be able to share some of our recent work trying to develop effective immunotherapies for patients with cancer. So I'd like to take a few minutes, because I know the audience is somewhat diverse, and just put cancer in perspective as it impacts on life in this country. You can see that there are about 600,000 deaths due to cancer in the United States each year. The overwhelming majority, 90% of them, are from the solid cancers. And given the incredible incidence of cancer, 1 in every 2 to 3 Americans now alive will develop an invasive cancer. And actually, 1 in every 4 or 5 Americans now alive will die of cancer unless we develop better treatments. Now, we have surgery, radiation therapy, and chemotherapy, which will cure so that patients can go on and live their normal lifespans. A little bit over half of all people who develop cancer, most of those are, in fact, cured by surgery, sometimes in combination with chemotherapy and radiation therapy. But this incredible death toll in the United States represents a major health problem that requires effort. Now, one of the secrets of modern oncology, it's not supposed to be a secret, is how effective our treatments are for patients with solid cancers once patients develop metastatic disease. So I would ask the students in the audience, but I won't call on you, how many different cancers, solid cancers, when metastatic, can be cured by any systemic treatment? So if you develop a cancer and it spreads from its primary site and cannot be cured by surgery, that is oligometastatic disease, how many cancers that we now see can be cured? Well, here are 2. 1958, methotrexate, for some reason we still don't fully understand, could cure patients with widespread choriocarcinoma. 1977, platinum derivatives were used to treat men with germ cell tumors. Lance Armstrong, even if you have brain mets, lung metastases, they can be cured. And what other solid tumors, besides these 2 rare diseases, choriocarcinoma and cisplatin, can be cured once they metastasize by any systemic treatment? Well, the answer is none, with the exception of these 2 diseases, melanoma and renal cancer, that respond to immunotherapies. And interleukin-2, which we introduced in 1985, caused complete regressions in small percentages of patients with melanoma and renal cancer that remain disease-free today. So if you develop a solid cancer, of the common cancers that comprise well over 95% of all solid cancers, and now I'm talking about the epithelial tumors, the tumors that start in the epithelial lining of organs, the rectum, the colon, the stomach, the pancreas, the esophagus, the prostate, the ovary, you name it. If you develop a metastatic deposit from one of those tumors, there is no curative treatment, no curative systemic treatment for those patients. And that's what accounts for these 600,000 deaths. Now, you read in the newspapers, a celebrity develops a cancer, I'm going to beat this. Nobody beats it. The death rate from metastatic cancer for the wide variety of common solid cancers is, what happened there? I hit the wrong button. What do I do here? All right, I'm going to need some help. So I need to get back to my presentation. I have to stay away from this.
Quick.
Good.
All the way back?
Yeah, go back. All the way back. All the way back. Oh. There you go. Thanks. Good. Yeah, good. Thank you. So, again, just to reframe the problem, the death rate from metastatic solid common cancers is 100%. And there's a desperate need for new systemic treatments. We can prolong survival, in some cases by many years with chemotherapy, but complete regressions will always recur, and patients will die of it. And that's basically the problem of modern oncologic therapy for solid cancers in adults. So the major challenge that confronts cancer therapy, not only cancer immunotherapy, is the development of effective therapies for the 80% of patients with metastatic epithelial solid cancers that cannot be cured now by any available treatment. Now, certainly there are advances in chemotherapy, targeted therapies, but permanent regressions are virtually never seen. So what can we do? to develop more effective treatments for these patients. Again, the combination of surgery, radiation therapy, and chemotherapy will cure over half of patients before they metastasize, but once they've metastasized, the story is completely different. So I want to talk about immunotherapy, which is the latest modality that has been developed to try to impact on this problem of the incurability of people with metastatic disease. So all approaches to immunotherapy can be put into these three categories. One can non-specifically stimulate immune reactions with the hope that, in fact, you'll stimulate immune reactions against the cancer. And that was interleukin-2, the first chemotherapy, the first immunotherapy ever approved by the Food and Drug Administration, but limited to patients with melanoma and renal cancer. There are new developments to inhibit regulatory factors, monoclonal antibodies like anti-CTLA-4 and anti-PD-1 that can be phenomenally effective in patients with melanoma, in patients with smoking-induced lung cancers, cancers that are known to be caused by carcinogens, bladder cancer. But in fact, when it comes to the common epithelial cancers, response rates are very low. Complete regression rates are in the low single digits. And this is new. Patients have been followed for only a few years, and so we have no idea of how prolonged these good responses will last. But patients, again, with the common epithelial cancers that I mentioned, do not respond to these checkpoint modulators. The development of cancer vaccines, a very exciting area of current development. As of now, there are no vaccines that can impact on the growth of these solid epithelial cancers, but a lot of work being done at this institution as well to try to develop effective cancer vaccines. The third area is the one that I'm going to concentrate on, and that is called adoptive immunotherapy, which is the passive transfer of immune cells to patients. You can identify a lymphocyte, which is the key effector mechanism of the immune response that recognizes a cancer. Can we isolate it, grow it, and then give it to patients to look for anti-cancer activity? Now, this kind of immunotherapy has some major advantages compared to more common systemic treatments. First, you can grow well over 10 to the 11 lymphocytes quite easily in vitro. So you can administer very large numbers of these patients. 1/10 the total volume of lymphocytes in the body can be replaced by transferring these cells. Because you have them in the test tube, you can develop T cells with high avidity for these tumor antigens. You can administer them activated outside the body, so they no longer react to the anergic and tolerogenic impacts of the in vivo microenvironment of the tumor. Because you have the cells in a test tube, you can identify the exact cell populations and effector functions that are necessary. And most importantly, it turns out, you can manipulate the host prior to the cell transfer to provide an optimal microenvironment to these cells, because the cells are out of the body at this point. And so you can manipulate the patient in ways with the radiation, with drugs, to eliminate these inhibitory elements, to increase the activity of the transferred cells in ways that you can't do if you're trying to give a vaccine or give an immune stimulant. And so this cartoon shows the general way that adoptive immunotherapy is done. And if you follow the excision of a tumor clockwise in this, we excise a tumor. We grow multiple fragments from that tumor, multiple cultures, to look at the diversity of reactivity. We try to select cells that have immune reactivity. We grow them to very large numbers, and then reinfuse them back into the patient following a lymphodepleting regimen, because it's essential that one first eliminate the regulatory T cells, myeloid-derived suppressor cells, IL-10 producing cells that can inhibit immune reactions. Now, I'm not going to talk much about melanoma, but we'll introduce the subject of melanoma because of the principles we learned that hopefully we can now apply to the common cancers that are the major killers that I have mentioned. And so this slide represents 194 consecutive patients that were treated with their own tumor-infiltrating lymphocytes. You put the tumor in culture, in interleukin-2, lymphocytes will grow out of that culture. And those lymphocytes, without further manipulation, re-administered to the patient following a lymphodepleting chemotherapy, will cause about a 55% objective response rate by classic oncologic RECIST criteria, about a 24% incidence of complete regressions, virtually all of which are durable and very likely curable. And you can see here the duration of those responses in months. Only 2 patients who had a complete response have ever recurred, now with median potential follow-up well over 7 years. And I'd ask you to notice that not only did only 2 of these patients recur, but in fact, all but 2 patients received just a single cell infusion. Cell therapy is a living therapy. You're giving viable cells that when they go in the body, circulate, when they see their antigen, they stop, they can multiply and perform their effector functions at the site of the presentation of antigen by the tumor. In fact, that's what an immune reaction is. These cells can expand over 10,000-fold in the first several weeks after they've been administered. So it's very different. It's a living treatment. It's not like surgery, close the belly, you're pretty much done, or the chest, you're pretty much done. Chemotherapy, most are excreted within minutes to hours of their infusion. And so this is a living treatment. It's a different kind of concept of therapy because these cells can survive as long as patients survive. We now found them in patients 20 years after their infusion. And so somehow these cells, these tumor-infiltrating lymphocytes, appear able to control the last melanoma cell, and you can see the survival of these complete responding patients. We're out here at 10 years. I made this slide in 2017. We haven't had any recurrences since then, and so these patients are very likely cured. Let me just show you 4 patients as an example of what cell therapy can do to make a few points. This is one patient that had a rapidly progressing cancer, and you can see in this CAT scan transverse cuts through the body. The growth of these tumors, we resected a tumor on day 0. We resected a tumor. We administered them on day 0, and you can see by day 34, virtually all of the cancers are gone. So 2 points. One, there's no relationship between the bulk of tumor and the likelihood of having a complete response in these 194 patients. When you have an immune system that can reject livers, lungs, you set off alarm bells and get lymphocytes growing inside the body. Large tumor volumes can be caused to regress. The second point is that it happens very quickly. You can see by day 34, almost all the tumors are gone, and this patient is now 15 years out and completely disease-free of this and other disease sites as well. So bulk lesions can be caused to regress, and it can happen quite quickly. Look at this patient who had his cutaneous tumors. This almost looks like a lymphoma response to chemotherapy, but you can see that this tumor disappeared in 12 days, and this patient is an ongoing complete responder over 10 years later. This very difficult case was a melanoma on the back of the skull, resisted multiple excisions, chemotherapy, radiation therapy. By 22 days later, it's turning black, and by 2 months later, it was almost gone, but it started to recur. This was one of 2 patients that required 2 treatments to achieve a complete response. And you can see here now, 13 years later, this patient is completely disease-free and lung metastases disappeared as well. So in melanoma patients, adoptive cell transfer can do quite remarkable things, not only in soft tissues, but the brain is not a sanctuary site for these cells. And in melanoma patients, you can see in this MRI scan, these lesions disappear completely over the course of 3 months. But TIL cells do not work in other tumor types. There's something about melanoma and a very large number of mutations that they have that make them quite sensitive to these lymphocytes. But we can conclude that adoptive cell therapy can mediate complete, durable, and likely curative regression in patients with metastatic melanoma. So that leads us to the question of how does this work? What do TIL actually recognize that enables the in vivo destruction of the last cancer cell? Now, we had an important clue in that the patients that underwent complete regression after the transfer of cells did not have any off-tumor toxicity. That is, there was no toxicity against any normal tissue. And that led us to explore what's the critical difference between a tumor cell and a normal cell, and that is all of the DNA mutations that are responsible for the oncogenesis of that tumor. Now, just a little brief excursion into an immunologic principle, which is absolutely critical if we understand the nature of immunotherapy, and that is how do lymphocytes recognize antigens? We want to try to identify what the T cell recognizes. We have to understand that T cell receptors on both CD8 and CD4 cells have two chains, alpha and beta chains, and what they recognize is an intracellular protein that's processed into small peptide fragments and brought onto the surface of that patient's MHC molecule, either class 1 or class 2, the transplantation molecule. And so it's this small peptide, which is what's recognized, but it has to fit into the groove of this patient's class 1 or class 2 cell surface antigen. And so for a mutation to be immunogenic, and most mutations are not, that protein has to be processed into small peptides inside the cell, and then by a variety of mechanisms that are quite complex and intracellular steering of these peptides, the peptide has to fit into the groove of that patient's own molecule, and therefore only rare mutations are going to have these properties. And so we developed a technique that we first described in 2013 and have been refining since, and I'm going to go through it very quickly just to give you a sense of what it can do. What we do is we take the tumor and an excised metastatic deposit or multiple metastatic deposits. We do whole exome sequencing and whole transcriptome sequencing as well of the tumor and normal tissue so that we can identify every exact mutation that exists in that cancer. Now, you remember it took 10 years, $2 billion to sequence a whole human genome. We can sequence every expressed gene now in my lab or any routine lab that has the equipment in about 8 to 10 days. You can identify every mutation, and by comparing to normal PBL, you know that they are in fact mutations very quickly. Bioinformatics takes another couple of days, but by 10 days you have all that information in hand. At the same time, you're growing lymphocytes out of the tumor deposit. And I'm just going into this technique because it's the technique that we've used for the entire rest of my most of my comments. What one then does is take every mutation and create a 25-mer peptide, either as a peptide or as a mini gene, putting the mutation in the middle. So in this case, we have a histidine that was mutated. Most of these are missense mutations, single non-synonymous mutations that alter amino acids. And this patient now has an altered mutated histidine in this 25-mer. And the key to this process was to put it in the middle of a 25-mer peptide, because now that histidine can be the first of a peptide that's 9 to 11 or 12 amino acids long. It can be the last one. But if this mutation is to be presented on the patient's own MHC molecule, it has to be included in this 25-mer. So what we then do is take all of those 25-mers together. Generally, we link them together into a tandem mini gene, put it in the patient's own antigen-presenting cells, and then incubate those with the patient's lymphocytes that caused a complete regression of the cancer. And so every mutation is being presented on the antigen-presenting cell, and we can now learn if, in fact, any of the T cells recognize the mutation. So this has huge advantages compared to other methods for doing this. You don't need to predict any peptide binding, and prediction algorithms are poor. Every candidate mutation and MHC locus is included in the screen, and you don't need a tumor cell line, which are very difficult to grow against the common epithelial cancers. Basically, this string of mutations is an avatar for the tumor itself. So let me just show you an example. But if you just would trust me that this can identify T cells that recognize mutations, and we've demonstrated this in hundreds of cases. Let me show you one example of a melanoma patient who had 71 non-synonymous mutations. That means a mutation that results in amino acid change. We constructed 10 tandem mini-genes by stringing 6 of these genes together and exposing it then to the patient's lymphocytes. And notice only this one tandem mini-gene encoding this individual genes, these 6 individual genes, was recognized in a co-culture assay based on interferon gamma secretion. And by testing each one of these 6 genes in a row, we can identify the exact gene that these T cells that mediated a complete regression, now ongoing and durable in this patient. And this turned out to be this KIF2C gene. It's involved in the stability of the mitotic spindle when the cell divides, and it was reacting against this kinesin family member gene that resulted in the regression of tumor in that patient. So now let me summarize. Utilizing that technique, let me summarize our results, not only first in melanoma, but then as we attempt to apply it to the common epithelial cancers. We evaluated 22 consecutive melanoma patients with this who had dramatic regressions of their cancer. They had a median of 318 mutations each. We screened about 4,000 mutations because we only screened the mutations that were being expressed based on RNA sequencing of the tumor. So there were 54 different immunogenic proteins that were recognized by the T cells of these 22 patients. Some patients recognized 2, one patient recognized 10 different. Interestingly, virtually all of them, in contrast to the epithelial cancers, were CD8 killer cells with very few CD4 helper cells. But quite remarkably to us, there was no sharing of antigens between patients. Every patient who had a complete regression was recognizing something different and unique to that patient's cancer. There were no antigens that were shared among two melanomas. So we've now done this. You can grow tumor-infiltrating lymphocytes in multiple cultures from virtually any tumor type, as you'll see. We started with our studies of patients with gastrointestinal cancers, all the way, as you'll see, from the rectum up through the esophagus. In 72 consecutive patients, they had collectively over 10,000 mutations, and we screened over 7,000 because those were the 7,000 expressed genes, and could identify 120 different mutations in the tumors of these 72 patients. And interestingly, if we looked at the T cells that could recognize these mutations, in contrast to melanoma, now half were CD8 and half were CD4 cells. So the helper cells can be quite active as well. Once again, in 120 different neoantigens across all of these epithelial cancers, everyone was unique, except for 2 patients that shared the same KRAS mutation and the same restriction element. KRAS is a very common oncogene. Well, we've now done this in 99 patients, not only in patients with the epithelial cancers that I've mentioned, but in endometrial, breast, lung cancer, ovarian cancer. And notice up and down the list, about 80% of patients with all of these histologies will give rise to neoantigen reactivity. By that I mean T cells. You can identify T cells that recognize the mutation in the tumor. These 99 patients gave rise to 197 different neoantigen reactivities, and again, 196 of them were unique to each patient's individual cancer, except for these KRAS mutations. And so let me show you an example of 4 patients that we've treated. The objective response rates in patients with widely metastatic epithelial cancers are the only ones that we treat, as we are attempting to improve our ability to identify the exact cell or cells that are necessary to mediate regression. Our response rates now are a touch less than 20% across all epithelial cancers. I want to show you 4 examples, and we're working hard now to try to improve upon it. But this patient had a metastatic cholangiocarcinoma, a bile duct tumor, a typical story of half the liver being removed, developed lung and liver metastases, received multiple chemotherapies, had progressive disease. We treated her with TIL that were just grown from the tumor. Well, that works in melanoma patients, doesn't work in the epithelial cancer. Then it didn't work in this patient. We were learning that as we did it. We then used this tandem imaging approach to target her unique antigens and identified, with the technique that I've mentioned, the identification of a single mutation in a tumor suppressor gene, ERB2 or IP, that attenuates downstream signaling and is highly involved in the oncogenesis of a cancer. We could identify subpopulations in this PAX analysis of cells that are almost 90% reactive with ERB2 IP mutation, administered them to this patient who had multiple lung and liver metastases. And all but 2 of her metastatic deposits went on to disappear completely. Only 2, and you can see some of these lesions that left scars, went on to completely regress. This lesion gone. This one going away. 3 liver lesions went away. Only 2 lesions persisted in this patient. Because we had increased the precursor frequency of anti-tumor cells in this patient, we gave the patient anti-PD-1, pembrolizumab, an antibody that can block inhibitory actions in the patient's tumor. And this one remaining lesion disappeared. This one shrank down, became PET negative. And this patient that was originally treated back in 2013 remains completely disease-free to the present time. We selected patients with some different histologies because this is not a histology-dependent treatment. Of course, we're targeting mutations, and every cancer has mutations. Virtually any cancer is potentially susceptible. This is a young woman who had an invasive ductal cancer, was ERPR positive. These don't respond to any checkpoint modulators. She had been through multiple different chemotherapies and hormonal therapies, as you can see on this slide. Came to the NCI in December of 2015 to receive her cell transfer. In that case, we gave it along with this antibody to inhibit negative reactions. And she's now in an ongoing complete response of multiple metastatic deposits, 39 months, over 3 years later. We just reported that about 7 or 8 months ago. Her T cells recognized 4 random antigens. These are random mutations that are being recognized, random somatic mutations that are being recognized by T cells. These are not driver mutations. And she underwent a dramatic regression, again, now lasting over 3 years. You can see this lesion, as so often happens in patients who recur following mastectomy, that was beginning to grow through the chest wall that disappeared completely, multiple liver metastatic deposits that disappeared completely as well. Here's a patient with a cervical cancer who had a terrible disease. She presented with a fungating cervical mass into her vagina, lung, intraperitoneal metastases, had radiation and chemotherapy, underwent surgery. Nothing was preventing the recurrence of these metastatic deposits. She came to us in March of 2013, received her own tumor-infiltrating lymphocytes that were highly directed against her mutations, and she underwent a complete regression of all of her disease, including relief of a urinary obstruction, and remains disease-free now over 5 years later. And here's an example of lymph nodes that disappeared, this abdominal mass which disappeared, multiple intra-abdominal masses. That tumor was obstructing her ureter. She had a urethral catheter that we could remove when her tumor disappeared, and she's again remained disease-free. The last patient I'll show, a patient with colon cancer, taught us something new. And this is a woman who had an aggressive sigmoid cancer. It was invading her bladder. She presented with multiple lung metastases, underwent a resection, including partial cystectomy, radiation therapy to that area, chemotherapy progressed, developed lung metastatic deposits. We harvested 2 to grow TIL cells and try to find the cells that were recognizing mutations. She had 61 somatic mutations, including a mutation against the KRAS oncogene, one of the two that were shared. And so again, we made tandem mini-genes. She had 61 mutations. Only one of her tandem mini-gene constructs were recognized. We could then deconvolute it and show that she was recognizing the KRAS G12D antigen, but not wild type. And in fact, in all of these cases, we recognized only the mutated peptide, but not the wild type, non-mutated peptide. Now, this patient then had 7 lesions in her lung, 6 of which regressed completely and have stayed away, but one of these lesions grew. You can see here this lesion 3 is the only lesion that was growing, as all the rest of them disappeared completely. This lesion went on to disappear completely, as did this. So we resected this one lesion, and the patient has remained disease-free almost 4 years later. But why did this lesion resist treatment? Well, when we did a copy number analysis, looking at each one of the chromosomes, it turned out that chromosome 6, which harbors all of the MHC alleles into which the peptide has to be presented on the cell surface, had lost one chromosome, one of the chromosomes of chromosome 6, which is this one. And in the absence of chromosome 6, that tumor could not present antigens because there was no MHC antigen upon which to place the peptide. And by resecting that one lesion, apparently, because the patient hasn't recurred, there weren't other micrometastatic deposits that survived as well. And again, we're continuing to follow these patients who have ongoing responses. So the checkpoint modulators, which are very active in patients that have very high numbers of metastatic deposits here, melanoma, as I've mentioned, smoking-induced lung cancer, bladder cancer, we can now see immunotherapy being extended to patients of the colon and rectum, patient of the cervix that I showed, patient with liver disease and breast disease as well, and we're working daily to try to improve upon these treatments and extend them to other treatments. So this would lead us to 2 hypotheses. One is that's the recognition of random somatic mutations that is the final common pathway that explains cancer regression for virtually all immunotherapies that can work. Interleukin-2, anti-CTLA-4 has been shown to, again, react against, at least there are several cases that have been shown to react against mutations. Anti-PD-1, the likelihood of PD-1 working is directly related to the number of mutations the patient has, and of course, these tumor-infiltrated lymphocytes. The second hypothesis is that any intracellular protein can potentially be a cancer antigen. We finally understand what cancer antigens are. We've searched for decades for shared antigens without being able to find them. It turns out that any protein can be a cancer antigen if it's mutated and processed intracellularly to a peptide that combines with that patient's own MHC molecule. And about 1 in every 70 of these mutated epitopes are neoantigens. Now, there's some good news in these insights. The first is that since all patients have mutations, of course, that's what causes the cancer, virtually all patients are potentially eligible for this kind of approach if we can improve it sufficiently. The bad news, however, is that it's the ultimate in personalized therapy, and the treatment will have to be individualized for every patient. We have to get cells from that patient. We have to attack a mutation that's unique to that patient. So it's hard to get more personalized than that. Now, I've concentrated on the recognition of these unique somatic mutations in intracellular proteins because that's potentially an approach for the treatment of many other cancer types. But there are 2 other major classes of cancer antigens that can potentially be targets as well that will be highly selective for the cancer. And that is mutations in driver oncogenes or tumor suppressor genes that can be shared among patients. And the 2 most common of those is the KRAS oncogene and the P53 oncogene. And there are others like PIK3 kinase that are shared somewhat. In fact, what's remarkable is how few shared mutations there are in cancer. You would think that cancer would have a very limited number of mutations that can cause it, but in fact, if you leave KRAS and P53, which are present in about half of all patients' cancers, and then BRAF is a mutation in melanoma patients, there are no other shared mutations that are higher than single digits throughout all of cancer. And it appears that it's the accumulation of these random somatic mutations that causes most cancers. KRAS, for example, which is mutated, and so we can target it, should not cause any normal toxicity, as you can see, is mutated in 70% of pancreatic cancers. And fortunately for immunotherapy, there are hotspots. Half of them are at this 12th position, another 30% at the 12th position with a different glycine valine mutation. And you can see the high incidence of KRAS mutations at these two major hotspots. So we could potentially develop T-cell receptors that recognize those shared mutations. And in fact, we begin to develop a library of these T-cells and T-cell receptors that we can put into a patient's lymphocytes that recognize many of these mutations, both recognized by class 1 and class 2 restriction elements. It's this library that potentially can provide an off-the-shelf reagent for the treatment of patients. We're also developing a library of reactivities against this P53 molecule present. It's the most commonly mutated gene in all of cancer. A little over half of all patients have P53 mutations, and virtually all of them occur in this DNA binding domain. And fortunately, there are a few hotspots within that give rise to cancer. And one of our very talented postdocs, Dr. Parisa Seda, is now accumulating a library of T-cell receptors that can recognize these P53 mutations so that one might be able to use off-the-shelf reagents of T-cell receptors that can be put into a patient's individual lymphocytes. I mentioned this fellow, incidentally. All of this work was done by fellows. And so I'm here presenting the work of many dozens of fellows that come to the NIH, often in the middle of their surgical residencies to do research. Some PhDs come for postdoc fellowships. And I haven't been mentioning a lot of their names, but this was all done by young fellows who come to the lab and work on projects under the guidance of our senior staff. Let me finish by mentioning the last example of cancer antigens. The main problem with most therapies is you can destroy the cancer, but you have terrible toxicities on a lot of normal tissues, which is why almost all of our effective immunotherapies now are targeting mutations. But if you have non-mutated proteins on the surface of cells on non-essential organs, and you can actually recognize them, and they can be recognized by antibodies, you can potentially now recognize these non-mutated proteins. You may be destroying the normal organ that they arise in. But as you can see, effective treatments can be developed based on this strategy as well. And the first one to be developed was the recognition of the CD19 molecule. And to do that, we have to use an unusual genetic construct to develop an effector cell. T-cell receptors have alpha and beta chains, two independent chains that recognize that peptide in the grooved MHC molecule. But Zelig Eshar at the Weizmann Institute about 10, 12 years ago described what's called a chimeric antigen receptor. Basically take the heavy and light chains of an antibody, join them as a single chain genetically, and then attach them to intracellular signaling molecules of a lymphocyte. And by transducing this construct into a lymphocyte, you can now give the lymphocyte the recognition capability of an antibody, a monoclonal antibody. And antibodies, in contrast to T-cell receptors, recognize the three-dimensional structure of a surface protein. And so we developed a retroviral construct. With retroviruses are the easiest way to introduce antigens into T cells. You can do it 80 to 90% efficiently, right? retrovirus. We constructed retroviruses that targeted this CD19 molecule, which is a molecule that's present on all normal B cells, B cell precursors, most B cell precursors, and almost all adult lymphomas and children with acute lymphocytic leukemia. And so we developed this construct and attempted to treat patients that expressed tumors that expressed CD19. And the first patient ever to be treated was this patient that we treated, a 48-year-old man who came to the NIH after a drastic history of chemotherapy, vaccines, checkpoint modulators, very aggressive chemotherapy. Virtually all of our patients have had aggressive chemotherapy or allotransplants before they come to us. He came to the NIH in May 2009. We had shown in animal models, which of course precede all of this work, I'm not talking about it today, showed that we could in fact cause mouse lymphomas to regress. Got permission from the FDA to do this for the first time. And this patient had an ongoing complete regression. Last time we saw him was in February of this year. He's ongoing now at almost a little over 10 years. You can see his large tumor in his mediastinum disappeared. This mediastinal tumor disappeared. Huge spleen returned to normal size. See these huge iliac lymph nodes, which disappeared. He had large lymph nodes surrounding his aorta and vena cava. Underwent a complete regression now, sustained almost 10 years later. Again, what you're giving cells is not a relationship between a complete regression and the bulk of disease. His bone marrow was infiltrated with this lymphoma. It cleared completely, but the price that we paid is that this treatment destroyed every normal B cell in this patient. And his B cells went zero and stayed there. Most patients will recover about 8 or 9 months because there must be some deep progenitors which were not eliminated. B cells disappeared at the time that his T cells recovered normally, as NK cells recovered normally. And so this kind of approach, which can be very effective in patients with lymphomas, is one that we try to repeat, but there are unfortunately very few cell surface proteins that are unique to non-essential organs, such as the prostate, perhaps the breast. But these treatments are being pursued. So we treated 28 patients with this. Almost 70% had RECIST objective regressions. Notice that half of the patients had complete regressions. And if you look at the duration of those complete regressions, only one patient has ever recurred, a little bit reminiscent of melanomas. The others are ongoing, and these are regressions durations in months. And if you look at the complete regressors, again, it looks a lot like that melanoma curve. And again, there's no relationship between the bulk of disease, and I'll show you just a couple of other patients. In this PET scan, every one of these deposits is a lymphoma deposit, which disappeared completely. This patient who had an entire right chest full of tumor, and you can see that on this PET scan, that disappeared completely as well. Kite Pharma then came in 2012, aware of some of our early cases, and performed a multi-institutional study, almost exactly reproducing our results. In 2012, the National Cancer Institute signed what's called the Cooperative Research and Development Agreement, enabling Kite Pharma to support our laboratory by donating it to the NCI. They started developing it in 2012. In 2017, they received FDA approval. And in October of 2017, incidentally, the fellow who came to my lab in 2012 was a former fellow, Ari Beldigran, who had been a fellow in the lab about 20 years earlier. Started Kite Pharma, and just 5 years after starting that company, Kite was sold to Gilead Sciences for $11.9 billion. And this treatment is now widely available throughout the United States and most of Europe, and plans to do this in China and India are well underway. Well, I'll conclude with this slide. I think we've shown that cell transfer therapy can mediate durable regressions in patients with metastatic cancer that are refractory to other treatments. The T cells that recognize unique somatic mutations can be found in tumor-infiltrating lymphocytes as well as circulating in the peripheral blood, but at about 1/100th the frequency in patients with common epithelial cancers, and that identification and targeting of mutations unique to each cancer or shared mutations such as KRAS or P53 have the potential to extend this cell therapy to patients with the common epithelial cancers. Thank you for your very kind attention. Yes.
So that's for the TMG that you where
We have a microphone coming. Hang on. What? Oh.
So the TMGs that you strung together one after the other, are there sequences in between them to prevent them from like being recognized by
what?
The TMGs that you strung together? Were there sequences in between them that prevented them from like overlapping with each other so that they're still recognized as, you know, a distinct mutation?
We didn't have to worry about folding of those because every mini gene is completely different than every other mini gene. And although they are connected together, and so there are some sequences which would not naturally exist in the tumor, none of those get recognized, but the patient's never seen them. It's an in vitro.
I see.
So, Dr. Rosenberg, so my question regarding we are studying the tumors in the nervous system usually highly immunosuppressive. Could you comment what would be the strategy for those tumors have a very little or few T lymphocytes or suppressed T lymphocytes?
Again, TIL do not work well unless the patient is lymphodepleted. That is, you eliminate T regulatory cells, CD3 positive cells. You eliminate myeloid-derived suppressor cells. These can inhibit reactions. Eliminate IL-10 producing cells. And so one can overcome many of the immunosuppressive properties of individual tumors, such as nervous system tumors, by that lymphodepletion, which you can only do if you're going to treat the cells that have already been removed from the body, of course. What makes adoptive cell therapy such a unique approach to treatment.
What do the partial responses tell you? Like, have you been able to look at what their T cells are looking at? Like, are they unfortunate in that they just don't have mutations that fall into class 1 and class 2? Or
able to find mutations about 80% of all patients with resolved histology, so that we're getting better at it. But the tumor evolves as it grows. It loses some mutations and it gains some mutations. And so it's absolutely critical for the further. Oh, I'm sorry. I'm sorry. I'll just stand here. I like to wander. I'm a wanderer. Yeah. And so as these tumors evolve, they gain mutations, they lose mutations, and that makes it critical to attack more than one mutation at a time, so that even if one mutation is lost, others might have appeared or others might remain, which is why, for example, in that breast cancer patient, we had to target 4 different antigens. And presumably the partial responses that we get mean that we haven't attacked all of the cancer deposits, some of which might have lost expression of the antigen as the tumor grew and metastasized. So I think it's critical, as is the case in combination chemotherapy, that you attack tumors from multiple directions and multiple mechanisms. Yes?
At the back.
Yeah.
Thanks.
Dr. Rosenberg, thank you for such a fantastic talk. I want to ask a question that's a little bit different. In thinking about sort of molecules and mechanisms, it's easy to get involved in the details and lose the breathtaking scope of what is happening, and the fact that these tumors were untreatable 10 years ago. And so the question I want to ask is thinking about sort of the vision of where this is going and, in a way, hope, right, that these are solvable problems instead of terminal diseases. What are your thoughts on going forward, and what are your thoughts on starting out with a vision, and how does that evolve over time?
So the vision we had originally was to see if we could find T cells that could actually recognize cancers and not normal cells. And as we did that, new problems arose. And this idea, this concept that every cancer has a unique mutation that is immunogenic made things far more complex. And as opposed to developing a pill or a single infusion for treatment, this is highly personalized and can be quite complex. Pharmaceutical companies don't mind spending $500 million to create the first pill for treatment, so long as they can make the second pill for $1, which is how pharmaceutical companies work. Here, here we're dealing with a different concept, because here you have to develop a new drug for every patient. And so when we started doing this, and in fact with the CAR T cells and CD19, and we were reporting these complete regressions back in 2012. It only lasted 3 years, but Ari Beldegrun, this fellow who had been in contact with me, was the first to realize that he could commercialize this difficult process, which is not creating one treatment for everybody, but an individual treatment for each patient, and started Kite Pharma. At about the same time, Novartis began those efforts as well, and then Celgene, and then Juno, a variety of other companies followed. Turned out Kite and Novartis got there first, but they showed that these highly individual treatments could be commercialized and performed in a way that they could reach large populations. I think that's true for these specific mutations as well. If you have something that works, you have metastatic disease which is incurable, and it works, the genius of American industry will find ways to make it available. I have every confidence that will be the case. And so the major goal is to find something that works, and then figure out ways to make it more widely available.
Okay, last question from Tony.
A question and a comment. Dr. Rosenberg, thank you so much for coming. It's a true honor to see this. You know, someone who sits through tumor board and sees patients with this widely metastatic disease and hopeless, this truly is mind-changing and gives us some hope. The question I have is, you've created T cells that are effective against specific targets, and you see these mutations. And the responders, it's amazing and it's fantastic to see the clinical changes. In the non-responders, is there something about their tumors, if they have T cells that recognize the mutations but don't work? Is the tumor exuding something that's changing the paradigm?
Well, tumors are very clever, and they have a lot of ways to avoid immunologic reactions. The simplest is to just eliminate MHC molecules, because unless you have an MHC molecule on the surface, there's no immunotherapy that will target it. And unfortunately, beta-2 microglobulin mutations are rare, but they do occur in cancers, which would eliminate the ability of a CD8 cell to work, but CD4 cells can still work. But there are other resistance mechanisms. It turns out for lymphocytes to mediate tumor regression, they have to be able to make interferon gamma. And if you eliminate the ability to make interferon gamma, most of these T cell populations will not work. And there are several mutations in the interferon gamma pathway that prevent interferon gamma secretion, which is another potential resistance mechanism. And so most of our work now is, in fact, studying the partial and non-responders to try to figure out why they're not responding, to see if we can develop ways around that.
So on that note, thank you so much for an outstanding talk. I can echo everyone's sentiments here. This is a little token of our appreciation, and thank you so much for coming.